Okay, I think we can start, and I would like to ask our experts, presenters to start the lecture.
Good morning, everybody.
Yes, Igor.
Okay, nice to be here for this third day of this training course. Just in a moment, allow me to share my screen for today's presentation. Can you see my slide?
Yes, it's okay. Yes, you can start.
Okay, I turn on the subtitle option. I hope that everyone can read the subtitles and that they are visible. Can you just confirm?
Yes, it's visible.
Okay, great. So in order for the translation to be done correctly, I will try to speak slowly today. And today's topic would be we will discuss about the repeatability and the usability. Before we begin, just short a review of what we have discussed about in the past 2 days. So we discussed about validation, what is validation, and what is verification. We discussed principles for validation, and we started with stage 1. This and about analytical. Yesterday we discussed about analytics. So today we will complete this stage 1 discussion about repeatability and reliable results. So what describes a reliable result produced from experimental measurement? And another question would be, how can we trust our results? Well, one definition says that an experimentally derived value is reliable, precise, and accurate. Okay, now let's. So accuracy. Accuracy is defined as closeness of measured values, diagnostic test results, to the true value. So this is a measure that will reflect the effectiveness or the appropriateness of the true value. Means that the status of the sample that we test, that it's prior to the testing confirmed by other valid methods. Precision of the measured values, diagnostic test results in this case, to each other. So we can say that the precision is more a measurement of a technical specification, or it's influenced by the technical specification involved in the process of the diagnostic test. Let's see a bit more, let's say, practically what this means and what can be the possible outcomes of the test. So in the quadrant A, we have results when we have all our actually outcomes or results are in the middle of the target. So in this case, we will have high accuracy and high precision of the results achieved by this diagnostic test. It will mean that our results are true, which means highly accurate, and they are very close, the results, the values are very close to each other, which will mean they are highly precise. in the B quadrant, this will show us that all the results are very close to each other, which will mean that the precision is very high, but they are far from the true value or the truly expected result. So this will mean that in this case, the diagnostic test give us a very low or low accuracy. In the quadrant C, in this case scenario, in this kind of values that are used or obtained by diagnostic test are close to the true value, but the values in this set are pretty much separate or different between each other, which means that this assay is highly accurate, but we have low precision. And in the last example, the example D, in this kind of results, we could say that the test is neither accurate nor precise. In all of these cases, in case that when we have a high accuracy of the test, we can say that the validity of our results is high. But when the precision is low, for example, we can say that we have low reliability of the performances of the people performing the test, or maybe some other components included in the system. So, in case of results such as presented in the B section, would indicate a systematic error. The C results will indicate a repeatability error. The results such obtained in the D quadrant will indicate systematic error as well as repeatability error. So in experiments, precision of a measurement method is based on the degree to which the repeated measurements under unchanged conditions are able to show the same results. So repeatability and reproducibility of a measured values describe the precision of the test. We will start first with the repeatability, and you can find many definitions of repeatability. Some include more and some fewer elements, but more or less they are all the same. So let's see one example, and in this case the definition says that the repeatability, or define the repeatability, that is the degree of agreement between measurements taken under certain conditions by the same observer or operator in the same laboratory. To illustrate this more clearly and to repeat it once again, so we have one operator performing the test using the same reagents on the same equipment on a same sample. And if you remember from the previous talks, we always refer to standardized or the referent sample material, to which we already know the amount of the analyte or the quantity of the analyte that should be tested. And all this should be performed in the same laboratory. Several repetitions of the same experiment should be performed over a short period of time. And at the end, we should observe and analyze the variation obtained in the results. So how to do it? Well, the precision or the repeatability experiments are designed to measure the random error of an assay over a predetermined period of time by multiple measurements of an aliquot derived from a homogeneous sample. So minimal variation in the repeatability is very important, and this is particularly important near the cutoffs that establish positive, inconclusive, doubtful or negative ranges or results, because the higher variability can result in incorrect interpretation at the end. So if we discuss about molecular assay, and in particular quantitative one like real-time PCR, the repeatability panel should consist of 5 samples and covering, and these 5 samples should cover the operating range of the assay. So for example, if we call this or label these samples like repeatability panel sample 1, which will be represent the sample with high analyte concentration. For example, it should give us CT value of lower than 23. The RPS2 sample will be the sample with medium analyte concentration, and the value of this sample should lie somewhere between 23 and higher than 23 and lower than 28 CT value. For the third sample in the panel, we would like to include a low analyte concentration, a sample with low analyte concentration. So this sample would be somewhere above 30 and below 35 CT. And for the sample number 4 should be the same. We can use maybe the replicates of the same sample, or we can include, which is better, another sample of low analyte concentration. And the fifth sample can come from the negative matrix, so the same matrix as the previous one, the matrix which is 35 CT. And for the sample number 4 should be the same. We can use maybe the replicates of the same sample, or we can include, which is better, another sample of low analyte concentration. And the fifth sample can be the negative sample derived from the negative matrix, so the same matrix as the previous one, the matrix which is suitable for the validated test, just to include, and with this sample in this panel, by this repetition, we can also obtain the results for assessment of the analytical specificity of the assay that we test. So all samples must be tested using the entire assay procedure, in this case meaning from the beginning or from the nucleic extraction procedure onward. So how to obtain this repeatability panel sample, how to compile one. So it is always the best if you can have a clinical sample in your biobank, in your laboratory, with different analyte concentration, and to use them for preparation of the repeatability panel. However, this sometimes or more often is not possible, and these materials, as we said in the previous days, is difficult to obtain, especially for some exotic diseases. And in such case, we can use spiking methods to produce these repeatability panel samples. So the best way would be if we can use a whole organism for spiking into the same matrix as the test samples, and by this to determine the limit of detection of the assay, and in such way to ensure the adequacy of the extraction process prior to commencing the real-time PCR. But in case we don't have or don't possess or cannot obtain a whole organism, we can use plasmid or genomic DNA of a known organism and to use it into spiking the suitable matrix. One way to prepare the matrix, the sample matrix for spiking experiment, is to pull the negative specimen to a sufficient volume and to allow aliquoting of at least 20 replicate sample for the testing that should be performed during this verification procedure. The samples prepared for the assessment of the analytical sensitivity, and these are the samples used to create the standard curve analysis, can also be used for constructing the repeatability panel as well. And we can, because if you remember from yesterday, for the standard curve analysis, we always use a highly positive sample, which we then dilute in tenfold dilution steps, and we can use those already prepared material and select one highly positive, one in the middle range, and two low positive samples. However, I would recommend that you use different samples to be more, let's say, sure that the performance of the test will include all the possible variants and to get more reliable results on your repeatability. So also it is recommendable, and it's if you don't have other reference samples, to use the proficiency test samples, which are also concerned as a reference materials, and they are very valuable material and can be used for spiking and production of the repeatability panel samples. The repeatability panel should be tested treating all samples and each of their replicates exactly like individual diagnostic samples. So accordingly, every replicate of every sample is subjected to an independent extraction. And just to show an example of what we mean by this, so if we have a one reference sample, for example, blood, which represents our RPS1 repeatability panel sample 1, which is a strong positive sample, we have a stock solution, and the stock solution represents a larger quantity of sample with known concentration of the target analyte. Then we make replicates, 3 replicates of the same sample, ABC, and we perform nucleic acid extraction on all these 3 replicate sample and run all the sample on the same plate and using the same instrument. So this is just an example for the first, let's say, sample in the panel, and this procedure, of course, should be performed for all 5 samples included in the repeatability panel. Repeatability panel. Sorry. Within assay or intra-assay, variation describes the variation of results within a data set obtained from one experiment. So it is determined using a minimum of triplicates, as we previously said, preferably 5 replicates, but at least minimum triplicates, of each sample in the repeatability panel in one assay run. So to assess the intra-assay variation or intra-assay precision, this is the all same synonyms, we must perform testing of the samples in triplicates in one run. So using the same equipment, same reagents, and run it in one plate by one person. Between run or inter-assay variations describe the variation of the results obtained from repeated experiments. So these variations are determined by testing the repeatability panel samples over several days. So this would be the table that you will get, for example, for your repeatability panel sample tested in one assay run on one day by the same person in the same laboratory. So you will have all your 5 samples. You have to do it in triplicates, as we said, and you should record the obtained results from each sample. And this would serve you later on to calculate your intra-assay repeatability or intra-assay precision. The inter-assay repeatability would be, to the first table, we will add another additional column, and you can see here day 2 results. So you record the results obtained on another day by the same person using the same equipment and reagent, and then put it in this table. Later on, from these results, from the first and the second day, and observation, actually the comparison of the results obtained for the same samples on the different days, you will obtain the inter-assay repeatability results. Another terms that we have to explain before we continue and toward calculation, explaining how we will calculate the inter and intra-assay precision or variability, it's a standard deviation. The standard deviation is a measure of how spread our numbers are, or specifically it shows us how much our data is spread around the mean or average value of the data set. So you have here the formula for calculating the standard deviation, both for population and the sample. By this, and the difference between this is that, and we usually will use in our verification study, actually we should use the formula for calculating the standard deviation of a sample, because it is a variation of a specific smaller, let's say, sample, unlike the population, which requires a large data set representing the wider population. But if you can see, when you practice, if you want to practice and see comparing the results outcome using both formula for the data set that you will have from the verification, for example, verification experiment or the repeatability and reproducibility analysis, you will see that there will be really small insignificant difference between the results obtained by both of these formulas. Another term is the coefficient of variation, and what this means, and the coefficient of variation is a ratio of the standard deviation to the mean, and show the extent of variability in relation to the mean of the population or the data set used. So you can, or the higher, this will mean that the higher coefficient of variation is, the greater the dispersion of results is. And usually the coefficient of variation is expressed as a percentage. So as we said, and you can see on this equation, it is the standard deviation divided by the mean value of the data set, multiplied by 100, of course, to obtain the percentage. So repeatability can be expressed as a coefficient of variation. Usually it's expressed with this value. And for continuous outcomes, the simplest approach is to estimate the standard deviation of the replicates of a set of samples representing the operating range of the assay, as we previously discussed. And so for assay where the standard deviation is proportional to the mean, the within sample coefficient of variation is oftenly calculated. So to see now the calculation for the intra-assay repeatability, as you can remember from the previous slide, we have this table where one operator tested all 5 repeatability panel samples in triplicate, and the obtained results is calculated by the previously provided formula for the coefficient of variation, and you will obtain the result for the coefficient of variation and the general in general for the real-time PCR, or for the real-time PCR, yes, the acceptable value of this coefficient of variation should be lower than 5%. For the inter-assay repeatability, the same table with the data from different days, results obtained for the same samples on the different day. Then for the sample number 1 and also for the other one, you have the results from the first replicate for the day 1 and day 2, then the second replicate for both days, and the third replicate. All these 6 results are calculated or taken into consideration when calculating the standard deviation, actually the standard deviation, and from that coefficient of variation. And you will obtain these results for all 5 samples, and it will give you, and by this you can evaluate or assess how is your repeatability. Of course, taking that your coefficient of variation shouldn't be higher than 5%. Reproducibility. So what is reproducibility? Reproducibility can be described as the ability of others to achieve results as close as possible to the original, using similar tools and materials according to the instruction given by the author in their own environment. So if we can compare, let's say, or try to explain more simply the repeatability and reproducibility, it would mean that if someone can score a goal, let's say from a distance, or produce something in some manner, the repeatability would be, can he himself repeat the same thing, and how close the results from the repetition will be, how he is capable to perform, to repeat the same results over and over and again. Reproducibility would be if somebody else would try to achieve the same results or do the same thing as the original author or the one performing first, and how close will his result be to the result of the original performer. So let's explain it with more pictures. For example, you have here Leonardo da Vinci, who painted Mona Lisa, and he is the original author who produced some product. Now other authors tried to reproduce what he has produced, and in this first case, for example, this picture, we can say that this reproduction is pretty close to the original. We can observe some differences maybe in the color, but more or less the shapes, the size, and everything else, it's very, very close to the original. So in this case, we can say we have good reproducibility. The second painter, for example, if we analyze this picture, we can say that, okay, it is pretty close to the original, but we can observe higher variabilities, both in shape, size, and colors. And depending on our intended purpose or the customers, let's say, we can maybe say, okay, this level of variance is acceptable, and we can sell this picture. Or if we have a customer with higher, let's say, requirements, maybe this will not go on the market. However, this third author, it's pretty far from the original. There are many variability in this picture, and to sell this picture, maybe we will require different kind of customers. So we can say that reproducibility is pretty low in the last example. Well, unlike these famous painters, the ultimate goal of the developer, so the producer of the diagnostic assay, is to design and optimize their assay so they can achieve high reproducibility rates. And the definition of reproducibility at the end would be that reproducibility is the ability of a test method to provide consistent results as determined by the estimates of precision when applied to aliquots of the same samples tested in different laboratories. In other words, we can say that reproducibility is a measure of the method's sensitivity to laboratory changes. And it could be influenced by moderate changes in the equipment performance or variation of the operator's technique and the lab environment. Reproducibility, as described, is a very important part of validation of an assay. However, for the verification purposes, in most cases, assessing the intermediate reproducibility should be sufficient and more achievable goal. So what is intermediate reproducibility actually? I find this term and it's very convenient for this purpose. And it says that intermediate, it's defined as that intermediate reproducibility is the term used to describe estimates of a reproducibility done in a single laboratory. So sometimes there can be confusion between repeatability and intermediate reproducibility because the definition previously as seen, it defines the reproducibility that it's the results in the of the testing of the same sample panel but in different laboratories. And just to see once again to compare the definition about repeatability and intermediate reproducibility would mean that, as we said, the estimates of the repeatability will require all stages of the replicated testing to be done by a single operator using the same reagent, using the same equipment on the same sample in a single laboratory. And all this repetition should be performed in a short period of time. And the intermediate reproducibility would be if more than one operator, if we include the second or the third one as well, performing the testing on the same samples using the same reagents, on the same equipment or on a different equipment, and as we said, on the same sample or maybe even on a different samples, on 2 different days or more days consecutively. So this calculation at the end of the variation in the results will derive the measure of intermediate reproducibility. To calculate the intermediate reproducibility, once again, we have the table, the result obtained for the repeatability panel samples by the first operator on one day, and on the same day we have another operator performing the test on the same samples. And of course record the results, the obtained results. Then we repeat the same procedure but on another day. So on the same panel samples, the test is performed by the first and by the second operator. At the end, how to calculate the intermediate reproducibility? on this example here, I present only the results and calculation for the intermediate reproducibility for the one sample, for the first sample, let's say for the RPS1 sample. So we have what we have. We have 3 replicates of this sample, and we have the results from the first operator on the day 1 and the second operator on the day 1, as well as the results for these triplicates by the first operator performed on the second day and the second operator testing these samples on the second day. So all these 12 results will be included as a data set to calculate the coefficient of variation. And of course, the acceptable level or the assessment of a successful or good intermediate reproducibility would be if the coefficient variation or CV percent would be lower than 5%. And how this was about the molecular test. And now to see, just to give some example or explanation or comparison how the reproducibility is expressed in ELISAs. So in ELISA, the reproducibility is significant to reported results. Why? Because it fundamentally describes the precision of ELISA's reagents and components working together to facilitate the antibodies binding to the target or the antigen binding to the target of interest and producing signal accurately to quantify its presence within a heterogeneous sample matrix from a population. So the overall precision of an immunoassay is defined by examining repeatability of reported results through examining variation between wells within a single run of a plate. So this will give us the intra-assay precision, as we said previously, or it will give us assessment of the repeatability of an assay. So usually this intra-assay variation or precision could be evaluated by comparing the results of the replicates of a referent or in-house standard sample, at least, or the minimum number of these replicates should be 10, and should be, of course, run on a single plate. For the sample used for this evaluation should be the value of this sample should lie somewhere in the middle of the range of values. So usually it can be +2 standard deviation from the cutoff. So not near the cutoff, but at least, let's say, 2 standard deviation above the cutoff. And the overall precision of an immunoassay is also defined by examining reproducibility of reported results through examining variation between wells in a different runs or trials, in another word, say plate-to-plate variation. All this will give us the inter-assay precision. So inter-assay coefficient of variation is evaluated by comparing triplicate measurements of each sample included in the repeatability panel sample obtained from independent runs on 3 different days. So to evaluate the reproducibility, it is recommendable to include different operators and different spectrophotometers or ELISA readers, if available, when performing these assessments. The repeatability panel samples should span the range of the standard curve. So as previously discussed and explained for the molecular test, so the same counts for the serological test. So we have to include 2 low positive sample, 1 in the middle range, and 2 in the high range, 2 highly positive samples. Now the difference between the acceptable criteria, for example, for these variations in the results in the serological test, in the ELISA, it says that the coefficient of variation should not exceed 15 to 20% for each point. meaning for the low positive, for the high positive, and the middle value positive sample. So unlike in PCR, when the coefficient of variation should be lower than 5%, in ELISA, this variation should not be higher than 15 to 20% in the reproducibility experiments. So this is one example, just to be more clear, for example, how we will interpret or how we will calculate or assess our intraplate precision or repeatability or intra-assay variation. We said we need a sample, reference sample, who should be in the middle of the range of the values, and we perform at least minimum 10 replicates, and let's say that we obtain these optical densities. If we plot that on the graph, we will see and calculate the mean value would be 0.618 OD, standard deviation would be 0.02, and the coefficient of variation for this data set would be 3.8%, which, and also it can be seen from the dispersion of the results on this graph, that we have good repeatability. So I think I didn't mention, I didn't see on my slides, but for the intraplate precision or the repeatability, the acceptable value for the coefficient of variation should be below 15%, 10 to 15. So for reproducibility it's a bit higher, but for intraplate precision it's the standard or the requirements are a bit lower. But in this example here we can see that it's way below 10%. And the same counts for the interplate precision, or when we assess the reproducibility. As we said, we have operator 1 and operator 2, two different operators performing the testing on the same panel samples of five samples done in triplicates, and we will calculate the standard deviation for all samples and the coefficient of variation for all samples, which will give us our, and for the one operator it will represent the value for the repeatability. But when comparing the obtained results for the same sample from operator 1 and operator 2, and then added to this one the results from multiple tests, so at least 3 different days testing the same sample panel, and we combine all the results for the same sample, we will obtain and calculate standard deviation and the coefficient variation. We will then get our values to be able to assess our reproducibility or interplate precision. And with this, I will finish. I think I'm finishing with my presentation for today. At the end, me and my colleague Vlado would like to cheer to all of you, and I think that it will be break after this presentation, and after the break, I will be more than happy, and we will be more than happy to reply to all the questions that you might have on this topic. Thank you once again.
Okay, thank you very much, Igor. Thank you very much. And as you requested, we will have now 20 minutes break. I would like to ask participants to prepare their questions and place them in the chat. After that, our experts will answer them, and we can discuss and comment. Thank you, and see you in 20 minutes.
Sorry, if I can just add before the break,
Yeah.
I think that might be because I have one, it was foreseen with the agenda, but of course we did not expect such big audience in this format. We prepared some set of let's say panel of data sets to exercise or to show practically this calculation. I presented now on my last slides for the ELISA, but I have one data set for the real-time PCR. So we decided to change, and then I will after the break, I will present, let's say, practically how this data set should be analyzed and what kind of results we will obtain. And after that, we will continue with the question and answer session, if that is okay.
Okay, this is excellent. Thank you very much. And then see you in 20 minutes.
Okay, thank you.
Okay, I hope everyone had a good coffee break. We have, of course, some questions, but for now, as Igor suggested, he will present some data set, and later on we will switch to discussions and questions and answers. Igor, are you ready? Can you hear me?
Yes, but during the break, we had a suggestion that we should first go with the question and answer session, and after that,
Okay.
I think that the data set will be provided to the participants along. It's a two-sheet document. On the first page there are data set, and on the second page there is a table in which they should include these values and try to calculate the intra-essay and inter-essay coefficient of variation. And then after, I don't know, half an hour or 1 hour, depends on their needs, we can go back and we can see if they have questioned, if they succeeded or not. And then we can try some of the participants to explain this. Or if not, if there is it's not really clear, then I can go and present them and show them how to do it.
Okay, excellent idea. Then we switch to questions. Ivancho?
Yes, that's a good idea. That Igor shared the template and then ask the participants to simulate in their laboratories and get some solutions. Get some participants to explain eventually how did they manage and have. It's a nice practical exercise. Okay, so it's a good idea. Thank you.
Okay, thank you. So the first question is from Sub-Institute. Can these methods and calculations be valid in RT lamp?
In RT lamp.
Lamp, yes.
Lamp PCR.
I think it's lamp PCR. Yeah.
It's not. It's the isothermal loop-mediated isothermal amplification. It's isothermal amplification of DNA RNA. So lamp PCR.
If I can add, real-time lamp PCR is a qualitative method to my knowledge. So it is interpreted as a positive or not, whether there is turbidity or not. So then for verification, you would follow the rules for qualitative methods. Igor, can you add something?
Yes, I agree. I completely agree.
Okay, the next question is from Nigeria, from NVRI, Dr. Tekki. What would be the answer to someone who asks the question, what is the repeatability of your result?
What would be the answer of the question, what is the repeatability?
Yes, what is the repeatability of your result?
Yes, well, this would be probably the question of the auditor during the accreditation process, for example, where you should demonstrate that prior to establishment of the assay in your laboratory or putting into the diagnostic purposes, that you have performed this verification at least, and that you, during this process, as we now explain, obtain the result for your repeatability. And the evaluation or the assessment of your repeatability is expressed through this coefficient of variation among different, in this panel of different samples. And if this if your results presented show that this variation is not larger than 5%, then you can say, okay, this is my proof that my repeatability is good and that my and that the test in my hands, in my laboratory, works according to the validation data from the producer.
Okay, the next question is from Indonesia, Harimurti. Do we need to determine the repeatability and reproducibility of commercial kit? How long the maximum time period of intra-assay repeatability testing between day 1 and day 2, since the storage time will influence the result?
It's pretty complex or huge question, but do we, if I remember all, so the first part is whether or not we should perform a verification or repeatability and producibility test for the commercial kits.
Yeah.
Yes.
This is the first one.
Of course. Yes, of course. We said that when we once, and this is the majority of cases, it would be that the laboratory will implement already validated commercial kit or assay. So for this assay, there should be availability of the validation data. So the user will know what is the prescribed specificity and sensitivity of the test, what is the probably limit of detection, and for us to be sure that the test will serve the purpose for which is developed and it's functioning correctly in our laboratory, we must perform this verification testings. So yes, if you have all the validation data, you should perform verification, which will include repeatability and reproducibility, as well as the analytical sensitivity or the limit of detection for some assays we discussed yesterday. And what would be how long before day 1 and day 2, if I remember it well. But this is not some strict, let's say, recommendation. So for intra-assay repeatability, actually for the test precision, it should be in short interval. For PCR, it could be even in the same day, so after a couple of hours, or it can be the next day. So it should be in shorter period of time, let's say, just to assess, to evaluate the repeatability. But for the reproducibility, it can be different time distance between the testing, because we would like to see the reproducibility of our results, the stability of the results and the whole system for the longer period. So maybe, I don't know, once a week or 2 times a month, or depending on the availability of the material and the reagents from the laboratory. But it should be at least 5 days apart, for example, for the reproducibility trials.
Okay, the next question is from Dr. Isha Teki, Nigeria. For ELISA, is interplate reproducibility same as intermediate reproducibility?
Is it intraplate reproducibility the same as intermediate?
Yes.
No, the intraplate repeatability, actually, it's the measurement of the repeatability as it says. So it is done by 1 or maybe 2 operators, but it will be evaluated separately. So to run at least, as we said, 10 replicates of 1 sample in 1 plate. So this will be the intra-plate repeatability. So the variations in 1 plate for the measurement of the same analyte, same sample. And intermediate reproducibility, as we said as a term, would be that probably most of the laboratories will not be, during the verification stage, let's say, of the assay, will not be able, or it's not really feasible that you will share the samples with different laboratories in order to evaluate the reproducibility. This is done by the producer during the validation stage of the assay. So, but you also need to evaluate what is the reproducibility of your results, given that probably in your laboratory maybe you will have two technicians who will perform the same assay, diagnostic assay, or maybe you have more than one machine. So you would like to see or to compare or to assess actually your reproducibility. And for this intermediate reproducibility would mean the reproducibility within one laboratory. So it will slightly differ from the definition in OIE for the reproducibility in the validation terms of when we speak. So this would be these repeated testings by more different operators on different days, including, not necessarily, but desirably, if you have different machines, let's say, different instruments. So the assessment of this variation would give you the intermediate reproducibility, and if it's good, then you can be sure that whoever performed the test on whichever machine, it will give you the satisfactory results, or as defined by a reference method.
Okay, the next question from Beyhan: how we can define the lab study in that two-person work at the same time and same conditions? Is it interplate reproducibility in ELISA? Does it cover repeatability? Again, long question with...
Yes. If two operators perform the test on the same panel of samples in the same time, but on a different plate, the result of each one of the operator will give you the intra. So in the plate, on one plate, repeatability, and if you compare both results, it will give you the measurement of intraplate, because it is now question of two different plates. So you will compare whether or not these two different plates will give you the same result. They might be from the same batch, but they can also be from different batches, for example, from two different boxes of, for example, for ELISA plates.
Okay, question from Sierra Leone. Is there any formula to calculate the coefficient variation for reproducibility of ELISA results when different samples are used?
Yes, the formula is given in the presentation for the coefficient of variation, and we said that the formula is actually simple. It just represents the relation between the standard deviation and the mean of the data set. So first you will have to determine the standard deviation of this of the results for one sample or group of samples, depending on the needs. And you will have this practically in the data set that I provided, that should be shared among the participants, and it will be nice to see if they can succeed. And at the end of this day, I will show and present how to do it. So yes, there is the same. It is the same equation, and it's actually consists of this. So your standard deviation for the set of results divided by the mean value of this data set.
Okay, so I see one more question from Indonesia. Thank you for the explanation. So for commercial kit, if some products, do we need to perform the verification in each batch or just once?
Yes. Now this all depends on the throughput of the laboratory, how often they use the test, how many samples they have. If they have a pretty frequent use, they can more or less, let's say, rely on the results of the internal control. Of course, you will always need to include in your testing, especially when you are opening or you're using new batch or new shipment, new delivery of the same reagent, you should perform verification using your standard panel of samples. So as we said, when you compile this repeatability panel samples, so two highly positive, two in the low range, and one in the medium, you should test these samples with the new batch, just to verify that the performances of this new batch is as is comparable or the equal with the one previously used. So yes,
Okay.
the question shortly is yes, you should verify it. It's good to verify each new batch of reagents that you receive in your lab prior to start commercial testing.
Okay, and probably the last question before we start this exercises. Can the repeatability and reproducibility of semi-quantitative assay be determined? If yes, how? If yes, please give example. This is from Isha Teki.
Yes. Well, actually, this because you need to calculate the standard deviation, the coefficient of variation, and for that you need numbers, you need data. So my opinion is that it cannot be done, but I'm open to what my colleague Vladimir would think about this. I would say no.
Theoretically, it can be done if you just take into account for example, of 10 positive samples. If one, let's talk about reproducibility. If one operator from 10 positive sample has 10 positive, that's 100% identification, and the other one, for example, has 9 out of 10 positive samples. That would be a case only in low detection limit samples. In that case, yes, you can compare two of them. For example, it will be 19 out of 20 detected by two operators. Then you can calculate a lower limit of confidence of that would be something like 76%, and the upper will be 98%. So again, there are formulas that we do not calculate manually. We have Excel sheets, and we put inside also for qualitative tests, and get what is the lower and upper level of confidence of these the reproducibility of these samples. The same is for repeatability. But the results are taken for each operator separately.
So we've got one more question. In case when two operators are involved in a reproducibility experimental set, shouldn't mean SD and CV be evaluated on the basis of the difference of the values obtained by each one and for each replicate? Example, mean of absolute value of result 1 means result 2, SD of such different, etc. This is from Dimitrius Papayanau.
I must try to locate the question.
It's in the chat at
yes,
9:27. Okay.
Yes. So if I understand correctly the question, when we evaluate the reproducibility, when we have two operators involved, 2 or 3 or more, and they're all testing the same set of samples, the same replicates, then you collect all the results obtained in the different from different operators from different days, and by this you would like to assess the variation in the obtained results for the same sample under different conditions, different operator, different plates, different days. So all this range of values that you will obtain for this sample for each because the replicates are the replicates of the same sample, and the replication or testing more than one let's say part of the same sample will also provide you with a variability. So comparing all the variations in this obtained result will give you at the end the final assessment of the repeatability or the reproducibility in this case when we talk about different operators and different days. So I'm not sure if I understood and if I reply correctly, to the question, but to evaluate this reproducibility for each sample, you should take into account and calculate the mean values and the standard deviation and the coefficient of variation based on the results obtained from all replicates of this sample in the study.
Okay.
If I didn't explain correctly, or if I didn't get the point, then the participant could try to reformulate the question, or
Yes,
to try to
I think
answer it.
they will. And the question from Morocco, I would like to get more details about how to prepare repeatability panel samples.
So more explanation how to prepare.
Yeah, how to prepare the repeatability panel of samples. So practically, I think.
Yeah. Okay. Okay. Let's try share the presentation once again, just not to talk into the black screen. Okay. So as I said, yes, here. So this should be for example, for the real-time PCR, this should be the composition of your repeatability panel samples. So you should have one strong positive, one medium positive, two low positive sample, and one negative. And for all this sample included in this repeatability panel, it is ideally, as I said, to be from, if you can have a reference material, for example, reference material which is strong positive, or if you have a clinical sample, so using the original matrix, of course, is it the blood or is it the tissue or whatever, that you perform some testing prior, maybe with different methods, different technique, and you confirm it's positive and it's strong, it's high positive sample, then you prepare enough homogenate and do extraction, and from this you will obtain your strong positive. Then if you have another let's say samples in your biobank which are low positive or strong positive from the clinical sample, it will be the best. But if you don't have, then you can prepare using spiking. You can prepare the same matrix, you can use the same matrix, and you can spike the appropriate amount of the analyte, concentration of the analyte in the sample, or you can dilute them after that. So you can obtain medium positive or low positive samples. If you don't have this, you can, as I said here, I think on this slide, you can use proficiency test panel. So if you have participated in the proficiency test, these are also counted as a reference material because their values are obtained or defined in the referent laboratories. So you usually will get at the end of the trial, you will get the values for this sample, and you will know that this sample is strong positive and it should be, for example, 20 or 21 CT value. So if you include this sample along with maybe some other that you have in your lab, and you can compare them, or you can optimize your samples according to this referent one, you can also use those kind of samples to obtain your repeatability panel of samples. Also, I said that for this experiment, for this repeatability study, reproducibility, you can use also the sample once you start with verification process. You can use the samples from that you use to prepare the standard curve analysis. So here you will have also, starting from the strong positive, you will have the middle range positive sample, and you will have low positive sample. So, but always always count to prepare enough amount of these samples so you can have for repeated measurements. So to have enough material from the same sample, because if you perform another extraction of the same material, for example, if it's tissue or blood, then this will not represent the same sample. This is already a different sample. So you homogenize, you prepare the enough amount of all of these samples that should be included in this repeatability panel sample, so you can have enough amount to realize multiple testing on multiple days. So for example, I give here one example. If you have a blood, let's say, sample of enough quantity, and you have determined the value of the target analyte, then from this sample you take and then make aliquots of appropriate amount, which will represent your replicates, for example, and then you perform the extraction on all these samples, because this will involve in the measurements also the influence of the extraction method. And then you continue with testing. I hope I replied to the question.
Yes, it was very clear and detailed. Thank you. I don't see any questions for now. Any more questions? And we shared with participants via attachment in MS Teams some data set, right? Igor, can you explain something about this data set, what we now share with participants?
Yes. Yeah. Yes, okay. Let me just once again try sharing my screen.
Then.
Okay, is it? Can you see my screen? Can you see the screen? Am I sharing?
Yes.
Okay,
yes.
Thank you. So this is the data set provided to you. You can see this is actually real life data. Let's say it's original obtained data from a repeatability and reproducibility study. And you can see here there are repeatability panel samples. You can see that the name of the samples, they are all in triplicates. So the first three, so this D0412, you can see there are three the same name. So this is the triplicate of one sample. It's strong positive sample. Then the next one, it's also strong positive sample. And this was just to tell you, these were the samples obtained from, and this is from lumpy skin disease PCR. And these are samples obtained by extraction of skin samples of clinically positive animal. And then we have included in this panel two samples from a proficiency test. So the next three samples are, as you can see here, low positive, medium to low positive, let's say 30 CT value, and then the last one, the fourth one, it's a low positive sample, so 33 and above. And as the fifth sample, we included skin sample of a cow which was negative. So it was a historical sample prior to introduction of lumpy skin disease in Europe. So we performed extraction, and of course we obtained negative results for this one. So you have operator 1 results from operator 1 on the day 1, and the results obtained for this same sample panel of sample from the second operator on the same day, and run in within one PCR plate in one run, so together, just two different operators. And then on the second day, again, the results from both operators for all samples. So with this sample on the second page of this document, you have a table, a template, let's say, in which you should put the results from the operator 1 and operator 2 for day 1 and day 2 for each of the replicates of the 5 sample, actually 4 sample, because the last one is negative. And here, as you can see, for example, I'm not sure if you can see my the mouse, my cursor moving on the table. Am I?
Yes.
Yes. Okay. Okay. So here you will have you should put the formula for coefficient of variation, and with this and this let's say value will tell you how is the intra-operator variability, actually the repeatability of the operator 1 and operator 2 for this first sample. And for all samples, you will get the coefficient of variation for each triplicates of each sample performed by the first and the second operator. Then on the right side here, you will have to include or you should calculate the coefficient of variation, which will give you the intra-assay variability, which will mean variation between the results for these triplicates by both operators. So here you should calculate the coefficient of variation from all these 6 results for this one sample, and so on for all others. And the same thing you should do with the data for the values for all these samples obtained on the second day. And again, you will put the same formulas in the specific fields, which will give you the repeatability of each operator and then the intra-assay repeatability. So at the end, on the lower here table, you will have to put in these fields, in each of these field formula, to calculate the coefficient of variation, which will represent the inter-operator variability, and it will include here in this field, it will be the formula coefficient of variation of the results obtained by the first operator for this sample. So for all triplicates, like this here and here, these results that will be in these columns, in these fields. So let's go back. It will be this result and this result. So you will take into consideration these 6 results when you should here include the formula and to calculate the inter-operator variation or coefficient of variation. So differences of the results obtained for this sample on different days by the same operator, and here for the second operator. In this field here, for the inter-assay coefficient of variation, you should calculate for the first sample, you should take into account all these results, so all these 12 results that you will have here from both operator 1 and operator 2. Or here in this data set, so you should take all these results here, all these 12 results, to be able to obtain the coefficient of variation for the inter-assay reproducibility. Was this clear?
Well, I think it's clear, but let's ask our participants if it's clear. Then how long time we give them to?
So I would say 1 hour maybe, because okay, I'm not sure if they can get or maybe I can send the formulas. Maybe they are in presentation, but or they can try to. I don't know what would be Ivancho, what would be a suggestion about the formulas for the standard deviation and the coefficient of variation. Should we provide the participant, or should I leave the slide with the formula? Share my screen with these slides so they can look at the screen and have the formulas in front of them.
Yes, I think you can share the formula, but we can also share. I will write right now here. One second, please. There are some of you are familiar with the Excel software, Microsoft Excel. So in Microsoft Excel, you have built-in formula. I will just write the formulas and then.
Yeah, I wanted to. Try, yeah, sorry. My intention was at the end of the exercise, depending on the final results, just to explain practically this on my screen, how to use this formula built in the Excel, just not to complicate. But okay.
Okay, show them later on, yeah.
Yeah. Okay.
Okay, so we give 1 hour. It means that.
I said, please, just for all the participants.
Yes.
just to inform them. Sorry for this, but
Should I
okay.
stop sharing my screen?
Yes, I will take over now. So it's you see, for those who use Excel, there is a built-in formula where you can simply write equal average and then mark the range for which you want to calculate the average. The same applies for the standard deviation. So it's STDEV, and then mark the range for which you want to calculate. And at the end, if you want to calculate the coefficient of variation, so it's the standard deviation divided with the average value that you obtain from your data sets. So these are the built-in formulas, but you can also calculate by a calculator, but it's a bit more complicated. So if you use the built-in formulas, much easier and better. Yeah, thank you. Over.
Thank you, Ivancho, for this clarification. And from now we have 1 hour, so we meet. We come back to our chat at 10:50. If you have questions, please place them in the chat, and our experts will try to clarify something to you. But try to manage within 1 hour, and then we meet again. Have a nice work. Discussions after exercises. I hope you all succeeded and were successful with your calculations. And now I hope our experts will take the floor to discuss, and also later on we have some questions to address. So Igor,
Okay.
the floor is yours.
Okay, so I should now present how it should be calculated, and then we will continue with the discussion and questions. Is that the plan?
Yes, I think so.
Okay.
If someone sent you these calculations, they did. No one.
I didn't. No, I didn't. I don't see any email. But however, we can ask somebody to explain, or I think that maybe it was the idea from the beginning. But okay, I can just explain shortly, and
But
then we will
maybe
see
we
if
ask
everybody done the
audience.
same.
Maybe we
Igor,
ask audience if someone sent
Yes.
it.
there is someone who sent at 10:28. Dr. Abadi from Syria
Okay.
sent a file with his calculations.
Okay, it is a shared file. Okay. I will try to access. Okay. I can share the screen. So this is the sent file. By the look of it, I would say it is done correctly, but I don't know by heart all values. And we can ask the participant to explain or to just to present briefly, and then I can do it for everyone, if it's possible. I will leave my screen shared.
So if participant can take the floor from Syria, how we need to enable him somehow, I think.
Yeah.
To make
yes.
as a presenter.
Correct. Maybe if Dr. Abad is there and can raise the hand, so it's easier to identify, we can allow him the use of the microphone. Yes.
Yes, we can hear you. Yeah, was it yeah, was it easy or was it clear, let's say, how to program the cells to calculate the coefficients of variation for all these data set? Yes. Okay, great. I think by look of the results, I think that everything was made correctly, but we will see now in real time presentation. Thank you very much. Okay, because I'm still sharing the screen, I can yes, move here. So we have the data set that all of you have received and you should work on. So we can just copy this table here for easy of use, and we can it here. But first, as I shared my screen previously during the exercise, this is the highlighted fields and the formula which you should use to calculate the coefficient of variation for each specific data set. So we will do it just again here in this field. Sorry, this one. Yes. Okay, just to present simply how it should be done. So for you to be able to program the specific fields to calculate for you, for example, as we said, we need to calculate the coefficient of variation. We just want to find what is the sum of the standard deviation for the particular set of results divided by the mean of this data set. So, as previously said by Ivancho, you have these functions already in the Excel. So if you can type, and here you can see already in the drop-down menu, you can choose which of these function you need. So standard deviation of the set of the sample, and then you should choose the range of the values for which you would like to calculate the standard deviation. So standard deviation, for example, for the sample number 1 for the first replicate, let's say from the first operator for the first day. So we choose these 3 results, the results obtained by the one operator on the same day for these 3 replicates, and we close the bracket, and then we divide. So we divide the standard deviation for this range of values with the mean or the average. So if we put here,
sorry, Igor, can you share the screen? I don't think that you're sharing the screen.
I'm not sharing the screen.
Yes, the screen is there. The screen is there.
Okay. So I should continue?
Yes, please.
Okay. So you choose the average function, and you again select the same range. So these 3 values. You close the brackets, and you put another bracket at the end, and then enter. So here now you will have the coefficient of variation for these 3 results. So this is the same thing that you should do for all other. So in our given example, Okay, this is already and okay, one another example for so okay, let's be more clear about it. So CV here, when we would like to analyze or to get the coefficient of variation to evaluate the variation in the intermediate reproducibility or inter-assay variation, which means that we will need to include all the results for one replicate done by all the operators on each day, or in this case, on both days. So again, we write the sum of the standard variation. For example, we will take the sample number 1. So we need standard deviation for all the replicates from all trials and during this verification experiment. So we mark these fields and divide them by the average. Again, we select the same data set. And here we will get, and this is, and as I said, usually coefficient of variation is presented as a percentage. So if you choose this field and then go up, and you can transfer it to percentages and then put at least one decimal place, so, or two if you want, so you will get more precise numbers. So in this case, the coefficient of variation for the inter-assay variability or the reproducibility would be 1.53. So we can conclude, we can say that our assay, the repeatability, precision of our assay, it's pretty stable, or it's good according to the criteria. So it's lower than 5% of variation between the results obtained from this one sample done in triplicate by different operators and different days. So here we have included all this in this field. You will get at the end of the course this Excel already with the included formulas. So you will need just to put the results of your results for the samples. Of course, you will change the name of the samples you use, and then automatically you will get all the necessary information. So if we use this table for this data set, so operator 1, the values, we will put it here. And
Igor?
operate. Yes?
Igor, there is a question from Dr. Abadi from Syria. He's asking, why do you add the sum function to your equation? In the previous table, the previous calculations you did, when
Yes.
you were dividing why do you use the sum function there?
Well, it's my I don't know, because this is, we want to calculate the, it's the I don't know, I didn't try actually to see if it will function without the, okay, let's try. It's, I'm just used to do this like in this way. So okay, let's standard deviation. Okay, you can then choose the same range. And then divide it by the average. So yes,
Same.
you don't need to do it, so it's just my habit. So you will get the same result even without the sum. So you can just, yeah, this way or this way, or this is the simpler way for you without the sum, but it doesn't change anything, as you can see. Okay, I will continue. So we will translate transfer the results for the second operator for the same samples on the same day, and we will obtain these results, which if we compare with the results shared by our participant, let me just see what, no, it was here. So 1% for the intra-assay variation, 1.31 and 0.87. And I think that, yes, we get exactly the same results. So the calculation was done correctly. And then we will continue just transferring or putting the values for all consecutive samples, and automatically we will get the results. Continuing with the next sample. Okay, so at the end you will have all the results for the intra-assay variation and inter-operator variation for each sample, and at the end here for each of the sample you will obtain inter-operator variation, coefficient of variation, as well as the inter-assay variation for this test. And what can we conclude if we would have such kind of results in real time? As I said, this is the real-time data from the verification experiment. So we can conclude in this case that in all, regardless of which parameter we will assess, we see that never we have any variance either in inter-operator or within operator performances or between the operator, and also comparing the both days of performance of the same results, which is greater than 5%, which is in compliance with our criteria for acceptance. So with this we can conclude that we have good repeatability and good reproducibility of our assay as implemented in our laboratory. So just here if when you, I didn't, because this is a bit maybe different from the previous one because the data values are a bit separated. So if you would like, for example, to include these values in this formula, you see that you separate it by this sign, so, because you cannot select them all at once. So you can go like, for example, let me show here. So standard deviation must include the equal sign, standard deviation, and then you select this, and then you select this one as well. And the Excel will give you appropriate labeling or appropriate signs, and then average, and then again you choose this one and then go and choose this one, and you close the brackets. As you can see here, you can get correct value. So just technically, practically, how you will do it, if you choose, let's say, this template, this format, and you put your results as you obtain them. So not like here, just to transfer the already prepared results. So you can have this table and you can perform your testing, and you can record the results once at a time, and then at the end when you finish with all repetitions, so you can here add the third day, for example, if you would like to have even better evaluation or assessment of your repeatability or reproducibility. So you can calculate then the values for all 3 days, and then again at the end you will have the results as in the lower table, which will indicate the variation in your results between runs and for a longer period of time. I think that this would be everything that I can think of to tell you about how you should work on these tables and this data set. And now, if there are any practical question or any question theoretical, we can continue with trying to answer them.
Well, for now I see only one question about your recommendation. What literature would you recommend for study on this issue? It's from Olga Yadovskaya from Russia.
Let's just try. Okay, just so we can have a subtitle. Sorry, can you just repeat once again the question? What is the literature that can be used for this? Was that the question?
Yes. What literature would you recommend for study on this issue?
Well, you can find a lot of let's say material on the internet for this topic, for the repeatability, for reproducibility. There are also requirement, actually the guidance in the OIE standard as well with regards to the, but as we said in the previous day, it's mostly, let's say, focused on validation. But what we have now on my slide, which you will have for the, okay, I can just go and find this slide. You have also reference with regards to the intermediate reproducibility, which will be the reproducibility within one laboratory, which it's different from the OIE definition that the reproducibility in sense of validation is the stability of assay or robustness of the assay when compared the results for the same assay, for the same sample in the different laboratories. Yes, here I think I have included the reference, the link to this literature. Yes, it's Jervis. It's statistical aspects of microbiological examination in foods. There is this concept of intermediate reproducibility, also with formulas and explanation about standard deviation, the coefficient of variation. But if you just search on the Google, there will be plenty of material and explanation about the meaning and the calculation. And of course, you can I think you can use this presentation, the material that we will provide at the end, just to simplify all these things and just give you the most important aspects, along with all the equations, formulas, how to calculate it, and also the practical examples.
Okay. Thank you, Igor, for detailed explanation. And then we had one question, but Michelle already replied it. I do not see more questions from the... yes, one.
Artem, sorry,
Yes.
can I just add an information for the participants? We are, as we mentioned at the very beginning, we are working on the development of generic standard operating procedure for all our partner laboratories on this topic, verification of newly introduced SOPs with all these parameters. So our experts are involved in the development, but we will have also several reference laboratories in included in the process of the development and review of the final SOP before approval. And this SOP will contain the theoretical part, a brief summary on the theory, what is repeatability, reproducibility, and why do we need it, and how do we calculate it. And then attached to that theory, we will have the Excel algorithm with the calculations. So all of our partners will receive the final SOP after we finalize it. I assume it will take few months, but the idea is to have a standard SOP verified by the reference laboratories, distributed to all our networks. So it will be, I think, the maximum of the possible harmonization obtained all over the world, among the veterinary labs, of course. Thank you.
Thank you, Ivancho. Thank you. And one more question from Isha Teki. Are these calculations applicable to ELISA as well?
Yes, of course. The formulas are the same, so you just have to put in your results. And for ELISA, you should use the optical densities values for that, and then you will, of course, get your calculation on that as well. I mean, you will get the standard deviation, and then you will obtain also the coefficient of variation. I think also I have included in my slide, not in this type of table, so comprehensive, so big, but I think that I show that this 10 replicates that I presented. Let me just, find this slide. It is this one. Okay, there will be no translation now. I'm not just familiar how to play it with the subtitles and not start from the beginning. So here is the data set, the optical densities for all these 10 replicates. This here just summarize. So you just imagine, I mean, in these fields here, it's an Excel table file, in which you will put the same equation, and but just selecting this data set, and it will give you the results. So in this case, for example, from this data set, from these optical densities obtained by repetition of the same sample, the standard deviation is 0.023, and this represent actually, if you go up until the third decimal, which will mean that this is the optical density, the standard deviation in optical densities from the mean value. So if you can see here on the graph, there are for all the samples, there are dots representing the optical density on the epsilon axis, and you can see that this is the mean value. It's 0.618, and you can see how, but of course this is the very close range. If we can, if we put here a bit to stretch this and to make it with higher resolution, it will be more further apart. But you can see how that this is the standard deviation from which the samples are close or differ from the mean value of this data set, and expressed in the coefficient of variation, just to assess the repeatability, you will have 3.8%, which for the, if you remember, for the serological assay should be from 10 to 15%. Let's say it's acceptable level of differences in the results. So yes, you can use the same table and you will obtain these results, and you can based on this result you can evaluate your repeatability and your reproducibility of your assay.
Well, we have few more questions. One of them is how to use, how does one apply the same principle of calculations to tests that give results in the form of titers or reciprocal of dilution? So how to apply this approach to data like 1 to 200 or something like that, I think.
I'm not sure I really catch the question.
This is a question from Priscilla Shabala at 11:17. How does
Yeah,
one
because-
apply the same principle of calculations to tests that give results in the form of titers of reciprocal deletions?
Well, depending on the numbers that you use for calculating the standard deviation, you will obtain the results. The end results will be in this value. So if you use titers, the correct values of the titers, so you will obtain these results only for the standard deviation, but the coefficient of variation will be expressed as a percentage. I'm not sure. I don't know if Vladimir can help or if he can understand the question and try to answer. I'm not sure that I can provide.
Yes, I'll try if I understood correctly. First of all, if the titer, for example, for Newcastle disease is 1 to 16 or 1 to 32, it is indeed dilution. The titer is expressed as 16 or 32. So we can just take these numbers and put into calculation. But the problem is that it is not really quantitative test, it is semi-quantitative. So if you compare two testings, if one is correct and the second operator should make a double mistake to have one dilution wrong. So it is really a huge mistake. But if you want to have more precise results, also OIE manual suggests that you make intermediate dilutions. They should not be, for example, if you have 10-fold dilutions, then you will make 5-fold or 2-fold or in between dilutions. For example, if the result is suspicious between 32 and 64 and you want to compare performance of these two technicians, then you can make dilutions between 32 and 64. I don't know, does it give the answer?
I hope it doesn't. If not, then the participant will ask again, maybe reformulate a little bit his or her question. Then the question is about SOP that Ivansha mentioned. Maybe Ivansha could comment if non-veterinary lab could also receive such kind of SOP.
I mean, you know, the deliveries of our program are not commercial, so we can distribute to other. I saw that El Fahme from Morocco, I think, posed that question. He is also counterpart in our national project, so we will distribute. The point is, within the VetLab network, we try to keep the things harmonized. So for the future, if other labs are not in kind of communication with us, they will not receive the regular updates and regular harmonizations. Additionally, but very, very important, in the VetLab, the whole point of the VetLab network, you know, of this networking thing is that people can help each other. So if you see each other every year through different events, you can help each other. Look, guy, Morocco can call Namibia, or Zambia can call, I don't know, Niger, you know. Hey, I need help. I cannot calculate this. I cannot calculate that. This brings a lot of knowledge between the network of the laboratories. So my point is that, yes, it is free of charge. We don't charge anything for that. You get it, but you are not connected in the continuous updates of the progress of the procedures. As you know, we have plenty of procedures. All our network partners, they know very well all the procedures we share with the liver, African swine fever, rabies, whatsoever, lumpy skin disease, sheeppox, goatpox. So all of you should have harmonized procedures. So the idea now with this verification of the assays, we want to have harmonized procedures within the network partners. Of course, verified by the reference labs. They are the ones who create the rules on what is verification and is it 20 times to be repeated or 50 times. They are the ones who calculate the statistics behind. So that's the only concern, but otherwise there's no problem for us to share. Thank you.
Thank you, Ivancho. Then the question is from Ren Terry. Is it accepted that CV different between two operator? Example, in the calculation operator 1 is 1.31 and operator 2 is 0.87. This question was made at 11:19.
Okay, so the question actually is it acceptable or is it okay, or why? If that was the question, actually, yeah, of course it's acceptable.
Maybe.
Yeah, it's acceptable. Of course, it should not only that it is acceptable. This is really minor variation. So the differences between the results obtained by one and the another operator, it's really small. So that's why we say the criteria for acceptance when we evaluate is it okay or is it not, it's that this variation shouldn't be above 5%. So if these variations between the obtained results are below 5%, then it's okay. So it's acceptable and it means good repeatability.
Okay, thank you. Well, I double checked our chat for more questions, but I didn't see any that I missed. I would like to encourage our audience to ask questions if they have. Well, we will maybe wait 2-3 minutes more for questions.
Hi Artem, maybe in the meanwhile we can repeat, because we have seen many people asking for the slides and the recording, that this will be available in the Zodiac info platform that will be launched soon. So all this information will be available for the participants. Thank you.
Thank you, Federico. Thank you. Okay, I think no questions anymore, and everything was clear today. Thanks to the audience for participation in this training session and for fruitful discussions and for participation in exercises. Well, again, someone asked for materials, and there are some comments. I believe our trainers could have a look at it.
Okay, I should stop sharing the screen because I'm not able to see the chat in this window.
Yes, because this suggestion for participants, if you don't place question mark, it's sometimes difficult to understand. Is it a question or a comment or
So this from Priscilla?
Yes.
Okay, so from explanation from the day one of the course, verification is checking if your lab can achieve the same data as that obtained from validation studies during method validation. Commercial ELISA will provide data on certificate of analysis. What of tests described in the OIE manual, example, complement fixation test for brucellosis? There is no validation data provided, so where do we start? At validation or verification? Yeah, we said during the first days that usually if you don't have all the information or the required information with regard to the validation data of some assay, you should perform validation, but not always you can find the required data within the, let's say, leaflets of the commercial kits or maybe on their websites. Sometimes they are pretty restricted or scarce information with regards to the validation of the assay. And in this case, you can ask I think you can first try asking directly from the producer to provide you with this information. And if this is not possible, then again, depending on your intended use of the assay, you should perform validation. And for this, of course, you will need to have a panel of reference sample, which we mentioned sometimes it's difficult to obtain. However, in this situation, I mean, when the CFT is in question, it is also good if you can have in your laboratory actually a reference sample to which you can try establishing the performances of the test, even though it will not be a full-scale validation. At least a limited validation of the performance of the assay can be performed.
Igor, if I may just complement.
Yes. Yes.
Just to complement, I see from the question, it seems that Priscilla is not informed about the situation with brucellosis. Brucellosis is a very well-defined disease. So each reference laboratory has reference standard sera harmonized between the network of the laboratories and expressed in international units. So you can order these samples and then calibrate or verify your CFT with that samples. If you do not get the expected result from the very beginning, what you need to do is to check the complement, check the antigen, and check the hemolysin, these three components. Also, the source of erythrocytes have to be always from the proven negative animals. So consider these things. So to balance the complement fixation is not an easy task. It needs some knowledge. This is number one. Number two, once you obtain the reference samples, this is 20 units, this is 30 units, this is 25 units, it's a very good idea to apply for a proficiency test, because in the proficiency test, they will send you a panel of samples with different levels of positivity. And when you titrate them in your complement fixation test, you should get exactly the same result as expected in the reference laboratory. So once you achieve this level, that your results match with the expectation of the reference laboratories, you can say, yes, my CFT is well balanced and well verified. So it's, as a task, is relatively easy because there is no much of statistics that you see here on the Excel file. But as a technical implementation for the technicians to titrate the complement, the hemolysin, the antigen, the erythrocytes, washing of the erythrocytes is quite a task. So try that and you will see it will work. It's not a magic thing, but it needs some effort. Yeah, thank you.
Thank you, Ivanchi, for clarification. And since we don't see any more questions, I think this is it for today. And thanks again to our experts and lecturers for fruitful lectures and discussions. And I hope to see you all tomorrow morning for the Thursday session. Thanks a lot and see you tomorrow.
Thank you all. See you tomorrow.
All the best. See you tomorrow.
Bye-bye.
Bye.